Good afternoon, everybody. Thank you for joining us today, and welcome to this PeerView CME event held in conjunction with the World Conference on Lung Cancer 2023, approved by the IASLC. My name is Dr. Stephen Liu from Georgetown University in the U.S., and today we're going to talk about shattering the limits of conventional therapies, reshaping the future of lung cancer treatment with TROP2 targeting ADCs and rational combinations. For this one-hour program, I am joined by two esteemed colleagues with a wealth of experience in this area. From Yonsei University College of Medicine in Seoul, South Korea, Dr. Sun-Min Lim, and from University of Las Palmas de Gran Canaria, Dr. Delvis Rodriguez Abreu. So, welcome to our two co-panelists, and I want to thank you to everyone attending, both in person and to our virtual audience, as well as to PeerView for providing this educational session, and Gilead for providing the educational grant for this session. If you haven't already, please complete the pre-event survey. You can access that using the QR code on the flyer that was on your seat. That will allow us to better assess the program. You can also use that program to ask questions and interact with our panel here. We'll answer those towards the Q&A session at the end of the program. For more, you can visit us at peerview.com/23trope2, where you can download slides and practice aids on this topic. Our goals for today are to augment your knowledge of TROP2 and TROP2 targeting ADCs or antibody drug conjugates in lung cancer, to improve your awareness of the monotherapy or combinatorial treatment strategies with these antibody drug conjugates in lung cancer, and some of the emerging clinical data, some of which will be presented later at this meeting, and to learn how to integrate TROP2 targeting ADCs into the treatment plans of appropriate selected patients with lung cancer and manage some of the adverse events. And so let's jump right into the discussion, talking about fundamental concepts of antibody drug conjugates. Now, I see some familiar faces in the audience that really are focused on lung cancer. Antibody drug conjugates are relatively new to the lung cancer world, but we use them throughout oncology and other disease types like breast cancer, like bladder cancer. We're going to start off with a general question. And so if you can scan that QR code, let's start here. Which of the following is not a main structural component of ADCs targeting TROP2, the ones that are currently in development for lung cancer? Is it the TROP2-directed antibody, the stable linker, the TROP2-directed payload, or are all of these vital components of these ADCs, or five? Not sure. You can key in your answer here. And that was, we use ADCs in other cancers. These are relatively new to lung cancer, so I think it's important we understand how these work, because there are some differences here that are going to be relevant to moving forward with patient care.
That's what we want to see.
So if you scan that QR code, you'll be able to participate in this poll. Should be able to show our answers here. Now, Sun-Min, this has become relevant now because we do have one antibody drug conjugate approved for the treatment of lung cancer, though not for TROP2. That really would be for HER2. Our focus is on TROP2 today. All right, this is a meeting for lung cancer, and I think as everyone in this room knows, the treatment paradigms for non-small cell lung cancer have dramatically shifted. They resemble almost nothing what we were doing 20 years ago. This treatment is guided by biomarkers, and the presence of an actionable alteration directs us towards targeted therapy, directs us away from immunotherapy. This has changed what we're capable of in lung cancer. We can see fast, dramatic, and importantly, durable responses. But the challenge here is resistance. And as good as our outcomes are, there's still a lot of room for improvement. We still face challenges with intrinsic resistance. Our response rates are not 100%, in many cases far from it. And acquired resistance, even when we get responses, they don't last, they're not forever. When we see resistance emerge, our options there, unfortunately, remain pretty limited. For all the advances we have, when patients progress on immunotherapy or targeted therapy, the majority of those patients turn to cytotoxic chemotherapy, agents that we're familiar with that have their own toxicities and, unfortunately, pretty low ceilings on what they can achieve. Question we have looking forward in the field: can antibody drug conjugates fill these existing practice gaps? Well, antibody drug conjugates, the class of medicines, I think it's important we understand how they work. And I'm going to turn it over to Sun-Min. Can you walk us through the structure of what ADCs are?
Yes, so thank you. So I'd like to walk us on how getting to know the antibody structures. So ADC is composed of three key structures, but most importantly, the antigen is required for the antibody to bind. So the ideal antigen is highly homogeneous expressed in the tumor, but not expressed in the normal tissue. And when the TROP2 antigen is expressed in the tumors, antibody targeting TROP2 can bind to that antigen. So the antibody has to have a very high affinity and avidity for the target tissue, target antigen, and needs to have a long half-life to stay in the bloodstream. And also the conjugation sites should have a minimal impact on the ADC stability and internalization and pharmacokinetics. So moving on to the next structure is the drug payload. This is a very important topic here. So as previously dealt in the question, the payload is not TROP2 directed. So the payload is the chemotherapy component that is highly potent, it's like a bomb in the cell, as a microtubule inhibitor or DNA damaging agents that are able to induce apoptosis in the cancer cells. Payload needs to be stable because we don't want off-target effects in the bystander issue, and demonstrates a local bystander effect. And as we'll be dealing with Delvis later, this is a maximized drug-to-antibody ratio.
Yeah, so we have the antibody that's targeting the certain antigen that we want expressed on the cancer cells. We have this payload that for now is a cytotoxic agent, though in the future it could be many different molecules, small molecules, immunoconjugates as well. Now this, what we're showing here, is a simplified representation. This is an antibody, this is a payload, but the details here are really important. That's one of the things we're learning about antibody-drug conjugates, is these small changes make a big difference. Now, Delvis, it's not as simple as this. We don't have an antibody with two little arms that are the payloads, right? There's a little more complexity to that, right?
Yes, Stephen. The first one is to say, are you sure that is a new kid in the block? or is it too old?
That's right.
No, it's right, because ADC is not new. In medical oncology, in breast cancer, is used right now with a lot. But as you say, as Sung-Min said, the payload is very important in ADC. But the amount of this payload is linked to the antibody is also very important, and there is quite of that. Here, these two antibodies are not anti-TROP, it's anti-HER2. But it's important to see, especially that some of them, the amount of payload, especially in the trastuzumab, the Rustecan, is DAR 8. And we will see this. And why? Because the higher the DAR, the higher the possibility of efficacy. But we need to be cautious with this, because maybe the higher the DAR, the higher the toxicity. And there is some preclinical study that also said that if the DAR is so high, the clearance in the liver could be high also. So we need to find the middle, this rationale, the exact rationale, to have a high efficacy with a low toxicity with this antibody.
Yeah, we live in an era of medicinal chemistry and synthetic biology where all of these details matter. These are really elegantly designed structures. They don't stumble upon these different ratios, as you mentioned, a balance between more drug, but sometimes that does affect clearance. And so all these details matter. Now, we've left the third part to last, maybe the least glamorous part of the ADC, but maybe the most important, and that's the linker. Can you tell us about the linker, Sung-Min?
Yeah. So linker is like a glue. So we need a linker to connect the payload to the antibody. So the linker, there are two types of linkers. So first is a non-cleavable linker, and second is the cleavable linker. So non-cleavable linker, if a drug has a non-cleavable linker, it should be internalized and should be degraded in the lysosomes only. So in contrast, cleavable linkers can be degraded under acidic conditions or could be lysosomal sensitive. So in theory, cleavable linkers can induce more bystander effects. However, most agents that are developed so far have cleavable linkers. So we'll need to see the toxicity profiles.
Yeah, so these linkers, we don't want one that's too weak that releases this topoisomerase inhibitor or DNA damaging agent, but we don't want it so strong that it doesn't release it at all. We're going to look at these ADCs in general, as we mentioned, there are a lot of details that matter. The antibody, the linker, the cytotoxic payload here. For the case of TROP2, largely topoisomerase inhibitors. We're going to revisit this polling question here based on what our speakers have talked about up front. Which of the following is not a structural component of ADCs? The TROP2 antibody, the linker, or a TROP2 payload? And so if you want to scan that QR code, key in your answer, we're thinking to move on to, I think, is a very important part, and that's going to be the mechanisms of action. And so if we can key in your answer here, we're talking about how these drugs work. And I think there's a short answer is maybe it's a little more complicated than we initially thought. So Delvis, can you walk us through a little bit of the antibody-drug conjugate mechanisms of action in general?
Yes, you're completely right, because it's not so simple. And my question is, do we have to refer to ADC as a simple chemotherapy? Because one of the mechanisms of action of ADC, of course, as you say, some mean with the payload is chemotherapy at the end. So one of the mechanisms of action is just due to the chemotherapy that is high potent, is more potent than the usual chemotherapy that we use to treat cancer patients. Or maybe it's also the mechanism of action related with the target, the antigen. I think it's both, and clearly it's both. First of all, the antibody link attach the antigen, and of course, can block the activity of this pathway in the receptor, but also internalize in the cells, and then can release the payload, and the payload in some case, like in sacituzumab, I will be talking about that, is 1,000 more potent than irinotecan, and we will talk about that. So we are giving the drug inside the cell with a high potency that can release, can destroy the cell, and then there's another mechanism of action, because when the cell is destroyed, this payload, this chemotherapy can damage the surrounding cell, even if they don't have the antigen. But there is another mechanism of action. We are talking about antibody, especially IgG1, that can have an active Fc region that can link, can activate ADCC, antibody-dependent cell cytotoxic, and can stimulate the immune system, and it's another way how ADC work.
So directly cell killing, the bystander effect with neighboring cells, but maybe this potential synergy with immunotherapy. And I think we're just scratching the surface on the potential for these drugs. We've got a lot of ADCs in development for lung cancer. On the left here, we see our HER2 antibodies, adotrastuzumab emtansine, in our NCCN guidelines in the US, but trastuzumab deruxtecan, our FDA-approved drug for HER2 mutant lung cancer. Others in development, we see for HER3, we have patritumab deruxtecan. On the far right, we have IDXD and MGC018 looking at B7H3 in small cell lung cancer, telisotuzumab vedotin for MET overexpressed lung cancer, and of course right there in the middle, what we think will have a major impact for non-small cell lung cancer will be our TROP2 directed antibody-drug conjugates, sacituzumab govitecan, datopotamab deruxtecan. They are fun drugs to say, so I encourage you to practice saying them. These are fun words. We've got a lot of them in development, a lot of different developmental strategies. This is just a list. You can download these slides for your own reference, but our focus today is on TROP2. And while there are other TROP2 ADCs in development, these are the two that are furthest along, that are most likely to impact our care. On the left is sacituzumab govitecan. On the right, datopotamab deruxtecan. Both of these ADCs targeting TROP2, both being developed in the non-small cell lung cancer space. Sacituzumab govitecan we know is approved for breast cancer, for urothelial cancer, no approvals yet for datopotamab. These drugs, though, are very different, and we're going to talk about some of these differences today. The differences are more than just the colors. We know Sassy is blue and Dato is orange, right? So beyond the color, there are a lot of other details that are different. So let's talk about some of these data. Let's start off with understanding what we know. What is the current patient selection strategy for TROP2 targeting ADCs in lung cancer? Are we using these drugs in patients with TROP2 expression by immunohistochemistry, by TROP2 mutation by NGS, TROP2 amplification, or is there no TROP2 selection? So if you key in a QR code, let's answer this question together. What are we using as our biomarker? The biomarkers for ADCs are a little complex. It makes sense that IHC would be used, but for HER2, we know that it's actually the mutation that's much more predictive of benefit than the expression. answers here, and it looks like most of us are saying that there's no TROP2 selection, but about a third are saying TROP2 expression by IHC. Well, Delvis, why don't you walk us through a little bit of biology of TROP2? What is TROP2?
Well, you're right, but TROP2 was initially thinking was more simplistic than it is right now, because it's a transmembrane protein that activate, especially not only the calcium, intracellular calcium signal that it was at the beginning we thought that, but we know that right now TROP is related with another adjacent protein like claudin and other that finally promote proliferation of the cell and evolution of the tumor cell, because it's not only this kind of addiction, but also P10, McKinase protein, and all those systems that finally produce a proliferation and metastasis of the cell. And especially, we know right now that TROP2 initially was related with the development in the embryogenesis. But right now, TROP2 is related, and we can find it in a lot of tissue, and not only in tumor tissue, but also in healthy tissue. And we have to keep into account, because we have to focus in the toxicity at the end, and we want to talk about that. So, the next. So we know also that as a transmembrane glycoprotein, TROP2 is highly expressed in lung cancer. In non-squamous, non-small cell lung cancer, it is around 64% of the patient, the expression of TROP2. But in squamous, could be up to 75% of the patient. And we know that the higher expression of TROP2 in the tumor cell, as you can see here, is a bad prognostic factor in lung cancer. And that's good. And we know it's good to know that, because we know that this population with this high expression can respond to ADCs in the future. We don't have currently recommended testing for TROP2, but we know that a lot of companies and pharma are doing that, because we need to identify a better biomarker also for ADC, and maybe we can discuss, Stephen, in the future.
We certainly want biomarkers, it's true, but because TROP2 is so highly expressed in lung cancer, especially in those that need the therapy the most, and because we are seeing activity, at least in models, in tumors that don't express IHC positive TROP2, right now these drugs are being developed sort of in an agnostic approach. Samin, has that been your experience as well?
Yes, I think so. So the TROP2 antibody, the antibody is not exactly defined. There are many different types of antibody. We don't know which is the gold standard as of now. So most clinical trials are derived non-diagnosed biomarker tested. Yeah.
That's right. So no biomarker selection yet. We'll work towards that. Here's a question for everyone. What strategies are under investigation to integrate these